I just want to thank everybody that's on for coming. This is a seminar series that we are organizing as part of the Analytical Resources Core here at Colorado State University. This is a large analytical services group, and we're trying to highlight some of the technologies and approaches that can be enabled through the instrumentation and expertise in our facility. We are calling on some of our external collaborators and experts to bring some of that knowledge to us when we feel that we need some more education in this area. And with that, I would like to introduce today's speaker, Dr. Pratik Jagtap. And when I call him a colleague and a collaborator, I would say that he's been extremely supportive, not only in helping us to learn about metaproteomics and data analysis, but actually been involved in helping us in supporting our grant that enabled us to purchase the instrument that is enabling us to do metaproteomics analysis. So through his letters of support, we have been able to secure funding to purchase this instrument. So I'm very excited to have Pratik present to us today. So Pratik received his PhD at the Center for Cellular and Molecular Biology in India in 2000 during his postdoctoral research in the Schuster Lab at Max Planck Institute for Developmental Biology in Germany, and then at the Andrews Lab at the University of Michigan. He worked on genomic and post-genomic analyses of Delavibrio, an endosymbiont bacterium. Pratik is currently a research assistant professor at University of Minnesota, and he's a co-lead of the Galaxy P project, which we'll hear about today with Dr. Tim Griffin. And he's involved a lot in development of analytical workflows in the emerging areas of proteomics and multiomics, and in particular, metaproteomics and proteogenomics. As part of this Galaxy P team, over the last decade, he's presented many oral presentations, posters, workshops, national and international conferences. And he's currently the chair of the ABRF Informatics Proteomics Research Group after chairing the Proteomics Research Group from 2018 to 2020. He's also on the executive board of the International Metaproteomics Initiative, which is involved in communicating and advancing metaproteomics research. So with that, I'm going to turn the table over to Pratik, and I look forward to his presentation. Thanks everybody for attending.
Thank you very much for your introduction, Corey, and for the invitation to give a talk here. Can you see the screen, the first slide here?
Yeah, we can.
So, yeah. So today's talk, I'm going to be talking more about the data that comes out of the instrument and how we can analyze it to get more information than just looking at single organism proteomics. That is usually what is used, mass spectrometry is used. So the area of research I'm going to talk about today is metaproteomics. And I'll try to go through the slides and explain to you how we have implemented some analytical workflows into a Galaxy platform and how we are disseminating this through various educational resources that are available. As Corey mentioned, my name is Pratik Jagtap. I'm from University of Minnesota. If you would like to know more information about me, there are quite a few links here that you can access. I work with Professor Tim Griffin, who's been shown here, who's part of the Galaxy P team, along with multiple developers as well as users and data analysts who help us to make this possible. So the talk is going to be divided into 5 sections. I'll start with an introduction to metaproteomics and functional microbiome analysis, and then also talk a little bit about implementing these workflows within Galaxy platform, and then moving on to quantitative metaproteomics, and then looking into one of the projects that we are working on right now from a data that has been generated from the FAMES instrument, the same or a similar instrument that you have in your core facility, to kind of try to go deeper into the metaproteome of clinical samples. And at the last, I'll talk a little bit about our efforts in education and promotion of these resources. So going on to the first section, most of you might be aware, microbiome research has taken a lot of, generated a lot of interest in the last few years about not only its presence in the human body, but its effect that it has on the human body or its health, right? So, for example, not only is it involved in diseases, um, but it can also affect your mental health and your normal health and other processes that are taking place in your body. It has been shown that the presence of the right organisms or the metabolites that are generated by these organisms can have a profound effect on various functions of your body. So there is definitely a lot of interest in gut microbiome, and there have been multiple studies, most of which use metagenomics method, use sequencing DNA. However, we think that we can go deeper into this by using mass spectrometry data, and I'll talk a little bit about that as well. Then there are quite a few other researchers who are also interested in looking at microbiome of various environmental systems. There is a great interest in looking at the microbiome that's present in soil, how it can affect agricultural produce or even growth of different plants or different plant flora. And then there is, of course, a lot of interest in ocean microbiome and other environmental systems. So there's a great interest in trying to understand not only the taxonomic composition, but also trying to understand how these bacteria or how these microorganisms affect either the health of the individual or the the ecosystem in general. So in general, researchers have used two or three different methods. The most prominent amongst these is metagenomics, wherein you basically start with isolating DNA from the environment or from the gut microbiome sample, if that's what you're studying. And the focus there is to understand what is the taxonomic composition of these of that particular environment that you're studying. So, and these methods, bioinformatic methods, have been developed and in general have been quite popular in terms of trying to understand what is the correlation of the taxonomy with the physiology that you're studying. In past few years, there has been some interest in looking into not only the DNA, or DNA, microbial DNA, but also looking at the microbial RNA and how that might give you a lot more information, not only about the taxonomy, but also about the functions that are expressed. And again, here, I just wanted to mention that this is looking at the RNA expression of these microbial samples. Our lab for last 10 years has focused on a method called metaproteomics. The difference between the earlier two methods is instead of looking at DNA or RNA samples, we look at the proteins that are expressed by these microorganisms. And hence we get a much better understanding of not only the taxonomy, but also what are the functions that are expressed by these by the microbiome in under investigation. And as you know, since most of the proteins catalyze most of the functions, it's actually getting much closer to the function of the protein as compared to looking at of the microbiome as compared to looking at the RNA that is expressed. We believe that when we do this, we should be able to unravel the mechanistic details of microbial interactions, not only with the host, but also with the environment by looking at what are the different proteins or functions or functional pathways that are expressed by the microbiome. So the term metaproteomics was introduced way back in 2004, almost 18 years ago. And the initial application was to look at simpler ecosystems and try to find out what are the organisms, what are the proteins that are expressed by the organisms that are present in an environment. The focus has very markedly shifted to now understanding the functions that are expressed by the by the microbiome instead of looking at the taxonomy, and hence we believe that there is a definite advantage that metaproteomics has in terms of getting to understand what different functions that could be expressed by the microbiome. So just to give a hypothetical example, if you had a microbiome with these two microbiomes with similar taxonomy, and let's say if they were subjected to dissimilar conditions, in this case, dissimilar nutrients that they're subjected to, it has been observed that dissimilar proteins or functions that are expressed, and that is what we are measuring using metaproteomics and to some extent metatranscriptomics as well. You could also have another example wherein you could start with a dissimilar taxonomy, and then you could subject these microorganisms to similar conditions, and hence they might actually express similar functions, although the composition of taxonomy is entirely different. So in both of these cases, metaproteomics is extremely useful, and we have started using that using some of the bioinformatics workflows that we have put in place. Just wanted to mention, way back almost 10 years ago, there was a manuscript which had, in a way, talked about why functions in microbiome are important. And in this particular study, what researchers had done was that they actually collected samples from various body parts from various geographical regions. So, for example, you would have stool samples coming from various geographical regions, and here is shown taxonomy. And as you can see in this stool sample, there is a vast distribution of the genera that are detected in these stool samples if you take samples from various geographical locations. So there is no there's not there's not much consistency in terms of the taxonomic samples. But when they took this data um, and used predicted functional modeling to look at what are the predicted functions that are expressed by these microorganisms, it was found that there is a lot more consistency when you're looking at the metabolic functions or metabolic expression of these bacteria. This kind of indicated, and that there is definitely makes sense to look at functions rather than taxonomy of a particular microbiome, because then you can set up a baseline, and if you see any perturbations, you should be able to detect that using functions as compared to looking at taxonomy. So just wanted to make a distinction in terms of microbiome analysis and functional microbiome analysis, wherein we are definitely interested in looking at the proteins that are expressed and hence the functions that are expressed by the microbiome. What I'll talk next is about how we started working and in the area of metaproteomics, and how try to tackle some of the difficulties that were associated with metaproteomics. So one of the few of the analytical challenges that one encounters with metaproteomics is that in single organism proteomics, for example, the search databases are relatively small. There's a single organism, and most of the proteins that you're going to detect are going to come from that particular organism. While in metaproteomics, the databases are usually very large because you are collecting samples, or you kind of concatenating all the proteomes from various taxonomic members of these particular metaproteome. And then there are quite a few homologous proteins, which kind of makes it challenging for you to detect, find out which organism does this particular peptide or protein come from. So in order to take care of this, there have been quite a few search algorithms that have been developed. And then protein grouping has to be done at the multi-organism level instead of at a single organism level. The identification statistics do get affected by large databases. The larger the database, your sensitivity of detection both at the protein and peptide level diminishes. And then your taxonomy is based on unique peptide identifications, uh, while you can actually detect functions of proteins, even though these proteins have been shared by multiple organisms. And I'll talk a little bit about that as well. So in order to kind of handle these multiple problems or multiple tools that are required for this, we decided to use the Galaxy platform. And it basically helps you to put in multiple tools and generate a workflow, and hence perform kind of an end-to-end analysis wherein you can provide an input and you get an output at the end of the workflow. So I'll just very briefly uh, give an overview of the methods that are used. So for example, if you generate a mass spectrometry data from your sample that you're studying, you can use a FASTQ file either from your metagenomics or metatranscriptomics data and generate a protein FASTA file. And then you match these spectra against the protein FASTA file to detect your peptides. And then again, there are quite a few database generation methods that are available. We also have multiple database search strategies available, including some strategies that handle searches for larger databases. And then once you identify these peptides, either these peptides are unique to a particular taxon unit, it could be a genera, it could be a species, or it could be an order, or they could be shared. And some of them might even be unassigned. So, there are these quite a few categories of these taxonomic analysis. But again, in our case, we would rely mostly on the ones that are detected as unique peptides to a particular taxon level, so that we can confidently say that this particular organism or this particular genera or this particular order was detected. On the functional level, you can assign these peptides to proteins, and then if these proteins have a function that is assigned to them, then you can kind of perform functional analysis either by using pathway analysis or using various tools that are available to kind of to try to understand what are the different gene ontology terms that these particular proteins belong to. So based on this, we developed a workflow, a very basic workflow to enable researchers to put in their MGF files or their mass spectrometry data. Then you search it against the protein FASTA file. When you search it, and then there are at least, I think, 2 or 3 different search methods that we have right now. But one of the most common ones that we use is a search GUI, which basically uses 2 or 3 or even more search algorithms to enhance the number of identifications that you have. And then you can use statistical methods or protein grouping and FDR statistics to detect the number of PSMs, peptides, and proteins. So once you have that, you can subject it to unipeptide analysis, which performs both taxonomy as well as functional analysis, and that gives you, functional outputs, gene ontology outputs, as well as you can detect taxonomy with either if you're interested in looking at the genera that are detected, you can set that up, or you can also look at species or other taxonomic levels. So, so this is one of the basic metaproteomics workflow that we have available, and there's also a basic tutorial that's available in the Galaxy Training Network. So, based on this workflow and modifications of this workflow, we have published quite a few papers. One of the first ones that we published was in 2012, and this was basically looking at human salivary supernatant. So, one of the postdoctoral researchers in Griffin Lab had published a paper on human proteins that are present in the human saliva. And what he had used was he had actually used an elaborate fractionation method to kind of expand the number of human proteins that are detected. And we decided to go back and analyze this data and try to find out if there are any microorganisms present in that. And I think we ended up finding something like 123 bacteria based on unique peptides that were assigned to them. And this was something that we published really early, almost 10 years ago. Later, we also looked at one of the studies wherein Professor Joel Rennie from University of Minnesota, he was interested in looking at the effect of sucrose on plaque dental microbiome and find out whether there are certain bacteria that are enriched, but at the same time, whether certain proteins that were differentially expressed in one case or the other. And I'll be talking a little bit about this particular case study while talking about one of the tools that we use to analyze this data. We also worked with researchers at the University of Minnesota. Professor Amy Skubitz had data from the cervical vaginal microbiome, and again, as with the salivary supernatant, they had initially analyzed it with for human proteome, but later they decided to, we decided to go and look deeper into this data, and we actually did find out that there are quite a few bacteria that we could detect through the same data set. And then we also worked with Professor Manish Bhargava at the University of Minnesota to look at the bronchoalveolar lavage fluid. So this is the fluid that's present in your lungs and can be an indicator of the the health of the individual who's going through any the health of the lung of the particular individual. And then just last year we published a paper trying to in the right in the middle of pandemic, we're kind of interested to see if apart from SARS-CoV-2 peptides, in clinical samples, do we find any co-infecting bacteria that could be present in the samples? And we did end up finding a few of them. And a few months later, there were quite a few reports of secondary infections occurring in various parts of the world, indicating that it is kind of important to study these secondary infections apart from, the finding out if there are um, indications of any COVID infection. So, these were all good in the sense this helped us to detect some of the bacteria. They also helped us to detect some of the functions that have been expressed in these. But we were really interested in to look at how how does the how do these proteins change? How do these functions change when they are subjected to different conditions? So for this, we started looking into quantitative metaproteomics. And for most of you who have been doing mass spectrometry analysis, you might be aware that there are two different methods, at least two methods that are used for uh, quantitation of mass spectrometry data. There are labs which use spectral counts, wherein they look at peptide spectral matches and try to correlate that with the expression of a protein. So let's say if you had a protein which was detected with um, 200 peptide spectral matches, or many number of peptides as compared to in other condition which was where there are only 10, then you would kind of go back and try to calculate uh, whether there was any change, any differential expression. However, there has been quite a few researchers who have started relying on looking at the intensity of the data, wherein you basically look at the precursor mass MS1 intensities and then try to correlate that with the the abundance of that particular peptide, and then you can make correlation. So we were very interested in looking at these, using these metrics so that we could quantify taxonomy as well as functions. So, just to kind of highlight on that, there are quite a few different measurements that one can do. And again, as I mentioned earlier, metaproteomics is what we are interested in. As some of you who work in this area of mass spectrometry will definitely see here, there is a lot of data here, and especially gets very complex, especially if you were to look at the chromatogram of these data sets, and you'll see that you're not only looking at spectra that are coming from a single organism, but from multiple organisms. So your complexity really goes up manifold. So um, while there are many taxonomic members of microorganisms that are known, there are still some undetected taxa. So there are going to be a lot of this spectra, or a lot of this information is still untapped. There are quite a few proteins that have not been assigned any functions. So if you look at any microbial proteome, you would find that 60% of the most 60% or maybe 50 to 60% of the genes are actually annotated based on homology or any experimental evidence. But there are others like 40% or sometimes even more which are unassigned, depending on how much that particular organism has been studied. So there is a need for quantitation of these uh, taxonomic or even peptides or functions, or proteins without any functions, just to understand how important these could be so that they could be characterized later. So with that motivation, we started working on a tool called MetaQuantome. One of students, a very talented student, Caleb Easterly, he had joined the lab, and we set out on using the peptides and their, the detection of the peptides, its MS1 intensity, the functional annotation that comes out from some of the software, as well as taxonomic annotation. And using this information, it could be kind of fed into this tool called MetaQuantome, and using various R modules, it would generate these various outputs such as data explanation, differential abundance, or cluster analysis. So this manuscript was published 3 years ago in Molecular and Cellular Proteomics. And what I'll show next is the use of this particular software so that we could use it for not only this, but other data sets that we have been working on. Just to give a very brief overview, apart from the detection methods that I talked about earlier, and let's just focus on the top part here, wherein you use search GUI peptide shaker, We also use quantitation tools such as there is a tool called Flash LFQ that comes from University of Wisconsin that we have installed in Galaxy. And once we have the outputs from that, that basically goes into this meta quantum tool, which has got multiple modules. And eventually, after statistical analysis is done, you get these visualization outputs from meta quantum. And I'll talk a little bit about that as we proceed. So looking at this case study that I mentioned earlier, sucrose-induced oral dysbiosis, this data set was this data set came from Professor Joel Rudney's lab. There was a mass spectral data was acquired from plaque samples, and these were basically 12 children from which who had dental caries. The plaques were collected from them, and then these were either grown in presence of sucrose or in absence of sucrose. So they were basically grown in bioreactors, and here is shown the pH values of the bioreactors as they are grown, and the points at which sucrose was introduced in the sample. So the green line here basically shows the pH of control samples, and the red line shows the pH or the average pH um, of the samples wherein sucrose was added. And as you can see here, sucrose is added at 2 points, and then samples were collected at almost like 50-hour time point. And then, so, there were 12 pairs of samples, one with as a control, and other wherein sucrose was added. And we basically took this mass spectral data and searched it against a large database, used some reduced database searching methods. And eventually the outputs that were generated were sent to MetaQuantum for analysis. So this is what came out of meta-quantum analysis. So if you look at the function, if you look at the principal component analysis that came out of meta-quantum analysis, you can see that the sucrose samples shown here in red, as compared to the control samples shown here in blue, they kind of separated from each other, right? But if you use taxonomy, the separation was not as good, at least if you look at the separation values that come out of this. And we have seen it not only for this data set, but other data sets such as cellulose biodegradation data sets and some other data sets that we worked on, wherein um, if you subject a particular microbiome to conditions, to a particular condition, and if you compare these, you see that when you use functional PCA plots, you see a lot more separation as compared to if we were to use taxonomy as a way to separate these outputs. What meta-quantum analysis also does is it also can tell you what are the differentially abundant taxa. It tells you what's the fold change as compared to, so this is sucrose as compared to the control, along with the Q value that is associated, kind of giving you an idea about what is the how much can you rely on this particular fold change. It also generates heat map cluster analysis, and as you can see here, there is a separation of these two, except for these one sample, the blue being control and the orange being the sucrose induced sample. One of the advantages of using meta-quantum is that not only does it help you to understand what are the functions that are differentially expressed, or protein or taxonomy that's differentially expressed, but you also can ask questions such as, if I'm interested in a particular function, let's say carbohydrate metabolism, what are the different bacteria that contribute to this? So for example, if you just look at the carbohydrate metabolism in control, what is the composition of taxonomy? What is the proportion of peptide intensity of the taxonomy that's present? And what is and how does it differ when you add sugar to it? And as you can see here, streptococci is the topmost member here, while that's not the case when you look at the control. And if you look at Fusobacteriaceae, which is present as a topmost member in the control, it is in presence of sucrose, it's definitely not the case. So and there's an explanation for that, and we discussed this with Professor Rudney, who basically told us that sucrose, in presence of sucrose, there is lactate that is generated, which results in increase in acidity, and hence as a fusobacterium, which does not thrive in high acidic conditions kind of falls down in terms of its abundance, and hence its ranking is low down here. While um, in control, the bacterium, the carbohydrates are utilized, and these generate weaker acids. And under low acidity, Fusobacterium thrives in mild acidic conditions, and that's why you see this as the topmost. So there are these questions that can be addressed. You can also ask a question such as, if I have a particular taxonomic unit, what are the different functions that are expressed by the taxonomic unit in two different conditions? I won't be showing that example, but that's something that Metacontrome helps you do. One other aspect that we are interested in, so we talked about spectral counts, and for Metacontrome, we use precursor intensities, or MS1 intensities. But we were also interested in looking at whether we can actually use data independent acquisition in metaproteomics and see if there are any advantages using that. So just to kind of give a very quick precursor to or introduction to this data dependent acquisition, which is the method that has been in use for quite a few years to acquire data. It kind of targets precursor ions and fragments them, and hence you kind of so here is shown precursor ions, and as you can see here, there is one small red one here, which is a low abundant peptide, while the green one is one of the higher abundance ones. So if you were to subject it to the DDA analysis, wherein MS2 spectra are generated, you end up getting these spectra. But as you can see here, the red one is missing. So low abundance peptides are kind of most of the times low or medium abundance peptides are missed out when you're doing a DDA search or DDA acquisition. While if you were to use data independent acquisition, wherein you are you you're not only focused on a particular single precursor ion, but you're looking at a much larger swath or much larger area to acquire the data. You end up basically acquiring this data and hence generate multiple MS2 spectra, and you kind of do it in a sequential manner, and you end up generating a really complex spectrum of sorts. So there are software that are used to deconvolute this data and find out what are the proteins or peptides that have been detected here. One of the advantages here is that you can kind of generate this digital map of that particular proteome, and also end up detecting these medium or low abundance peptides in these samples. And hence, this seems to give a lot more reproducibility and also a lot more quantitative, um, it's a lot more amenable to quantitative analysis as compared to MS1 data analysis. So we wanted to kind of look at this and see if this indeed was case. So we last year at ASMS we analyzed and presented two data sets. One was bacteriophage infection data set, and other one was a hydrothermal vent data set. And based on these, we found that if you were to compare the proteome coverage, so these are basically peptides that are present in 4 replicates, and you can see that only 57.7% of peptides are present in all 4 of these samples, while if you were to use DIMS methods, the coverage was much more impressive. We also found that there was a quantitative reproducibility. Shown here are these 4 replicates that we analyzed for the bacteriophage samples. There was a higher accuracy with the DI method when you're looking at the biomass that's present in these samples. And then there were far less missing values and a lot more information that we got from DIMS analysis, and far less missing values in terms of proportion as compared to DDMS analysis. So we think that DIMS could be a method that can be used for metaproteomics as well. It has been successfully used for single organism proteomics, and we worked with Sue Weintraub from University of Texas and Brooke Nunn from University of Washington, who had generated this data set so that we could analyze them. The next thing I wanted to talk about is one of the kind of workflows that we have been developing, and this has been a result of collaboration with researchers um, who have expertise in various, not only in sample preparation, data acquisition, and then for this one, we actually used a method called as the FAMES method, which helped us to go much deeper into the microbiome samples as compared to if we were to do it on a normal mass spectrometer. So just to kind of give a, a very basic overview of this particular project, we were interested in looking at cystic fibrosis samples from children, and we basically we had with our collaboration with researchers in Illinois, we had quite a few samples from the both the groups. So shown here are CF samples, and the L here stands for samples with low microbial diversity, and H stands for those with higher microbial diversity. And DC are samples from disease patients, but they did not have any cystic fibrosis signs. So based on this, we basically had these four kinds of samples. We generated a lot of data from the spectral data from this, which was fractionated, and then we matched these data set against a really large 16S RNA guided database, and that helped us to generate a much smaller database. And we searched it with three different pipelines: FracPipe, which comes from University of developers in University of Michigan; MaxQuant, which comes from Max Planck Institute in Martinsried in Germany; and SearchGuen Peptide Checker, which comes from Compomix, which is based in Europe. So using these three search algorithms, we identified proteins, and shown here in parentheses are the microbial proteins or microbial peptides that were detected. So we ended up detecting almost 2,300 microbial peptides. And I kind of forgot to mention this, but whenever we are working on clinical samples, the most prominent proteins or peptides that are detected come from the host human sample, and hence it is important for us to uh, detect microbial peptides. So the microbial peptide signal is really of a very small proportion, and uh, using FAMES data and fractionation and using these uh, database reduction approach, we could end up detecting quite a few peptides here that I have mentioned. So once we had these microbial peptides that were detected using these three methods, we use a tool called PepQuery, which is a really nice tool, and we have also implemented it in this Galaxy, wherein it can basically look at the evidence that whatever peptide that you have detected, there is enough evidence that this peptide, um, uh, there is enough spectral evidence that this peptide is not present in your reference sample. And in this case, we were comparing it with the human UniPro database, and from the out of the 2,292 micropeptides, we found 680 of these peptides had a pretty good evidence that these were indeed present in the sample. So, again, this is just the proportion of the number of peptides that were detected by each of these methods. And when we use PepQuery, which is kind of agnostic to any search algorithm, it basically looks at the peptide and tries to find out the evidence for that peptide being present in the sample. We ended up getting much lesser, but confident peptides that we got that were detected in either 2 or 3 samples, 3 of the search algorithms. So, using this method of multiple search algorithms, PepQuery, and then eventually using UniPep, which is used for taxonomy and functional analysis, we could kind of bring it down to a list of verified microbial peptides, which still give us a pretty complex taxonomic tree. And this particular table, which was generated by Catherine Doe, who is a student here at the University of Minnesota, we could find 3 different classes of these peptides. And again, just wanted to mention, these peptides had gone rigorous analysis to ensure that indeed these were present. There was good quantitation data available, and these are confident peptides based on the uh, search algorithms that we had used. There were quite a few peptides, almost 24 of them, which could be subjected to a particular taxonomy, which was interesting. Um, there were some others which could not be assigned to a particular taxonomy, but they were still interesting because some of these actually had some signatures which indicated they could interact with host cells. So we are very interested in looking at some of these. There were some of these proteins, peptides coming from proteins which were ambiguous in its taxonomy. And what I mean by that was, it was not very clear whether they actually came from microbial, where they are of microbial origin, or they had uh, some properties that indicated maybe that they were present from the host immune system. So we are very interested in looking at these. We also looked at the quantitation of these peptides, and shown here is an example of one such peptide from Pseudomonas, wherein you can see that they were present highly, were much more abundant in the CF samples as compared to the DC samples. On the other hand, we also found a few peptides that were detected um, in only the DCH samples and nothing else. So we are very interested in going ahead and doing some targeted analysis on these and ensure that these give us some markers for not only detecting infection status of the particular individual that one is working on, but also maybe find out if there are any host peptides or proteins um, that are differentially expressed um, when you are basically in fact when you have cystic fibrosis. So the big plan, and we are still in working on this project, is to have a panel of microbial peptides as well as human peptides, so that you could use them for targeted analysis and hopefully help in diagnosis of the state of the state of the patient that is under study, so that one can understand whether cystic fibrosis it should be treated as a cystic fibrosis or should be treated otherwise. And again, this work is led by Professor Tim Griffin, and we have been working on this along with Monica Crook, who's one of the members of the, who is one of the researchers at University of Minnesota. Lastly, with the time that I have, I'd like to just very quickly go through some of the education and promotion material that we have been working on. So metaproteomics has its strengths, as I've tried to um, uh, convey that, it can help you in finding out the functional composition of the microbiome. It helps you to understand the host microbiome interaction. It helps you to quantify biomass contributions. But again, there have been also problems in terms of how do you extract a protein? How do you start with handling false positives? How do you handle variability and abundance issues that come out from microbiome or functional microbiome analysis? So with this in mind, we were um, for last 5 years now, we have been actually holding quite a few tutorials, both in person as well as online, to introduce metaproteomics to new researchers. We also hope that this will make tools and workflows accessible to researchers and hopefully attract new talents and perspectives. And this is kind of in line with some of the other tutorials that some of you might have attended. There's a Skyline tutorial. There are summer tutorials in MaxQuant conducts, and I think Transproteomic Pipeline also has some workshops. So we hope that we'll have regular metaproteomics workshops as well. As I mentioned, for last 5 years, we have presented workshops at various venues. And if you're interested in looking at some of these, you can go to this link here and get more information as well as material that's available from these workshops. You can also contact us if you are interested in any of this material. So I just very quickly wanted to take you through one of the workshops that we held in India in last fall, and this was and again there are these links here which you can go to and know more about it. But just to give you an overview, this particular workshop was a week-long workshop, and in the morning there used to be talks by eminent researchers from all across the world. This was, since it was online, we could invite researchers from Europe, Australia, US, at the right time so that students can hear it. Then we introduced the researchers or students to training material, and then this training material were basically videos of step-by-step instructions so that researchers can could run these on their own. And then the participants ran these workflows based on these instructions. And then in the evening there was a Q&A session, and the next morning we used to have quiz sessions so that we could test how well the students had followed this. We did this for introduction to galaxy, metagenomics, metatranscriptomics, and metaproteomics, and we also collected their feedback, which is kind of captured here in word clouds that have been shown here. We also collected feedback based on what they thought about the quality of talks, satisfied with the workshops, and as you can see, there was it was a really fulfilling experience because not only did we enjoy the experience of teaching multiple researchers about this, but also I think they enjoyed a lot, as you can see with these reactions that we got from them. Almost 80% or more of them were very happy with uh, various features of this workshop. Um, so just to kind of come to a conclusion, we work very closely with the Galaxy Training Network. And again, for any of this information, you can go to any of these links to get not only step-by-step instructions, but also video tutorials and also access to some of the public servers that you can access these along with the input data if you want to learn about the tools that I mentioned here. We also work very closely with the Metaproteomics Initiative, which is a European um, proteomics association funded metaproteomics society, but is it covers all of the world, and we definitely I'm an active member of that particular community, and we are definitely looking forward. If people are interested, they should definitely join in if they want to either participate or learn more about metaproteomics. Well, the long-term goal is to have all of these tools available so that eventually that leads to your biological discovery either through online training or workflows that are accessible through various public Galaxy servers. And again, if you're interested in any of these tools, they're available on this either in the Galaxy Toolshed or on the Galaxy Training Network. And if you don't find anything there, or if you find something interesting, if you are interested in collaborating with us, or even have any questions about any of the workflows, you can also contact us on this link. Um, I'll maybe just skip this because I think I've covered most of this. I've talked about DD and DI analysis and quantitation, but we are very interested in working in this area. We're also very interested in using targeted metaproteomics to validate the proteins or peptides or functions that we have detected in our discovery workflows. And we are also interested in looking at how quantitation can help us to find more information about unknown proteins of unknown function. Um, with that, I'd like to acknowledge some of the researchers, Professor Tim Griffin, who's the PI on the Galaxy P grant. He is Galaxy P project. He's helped us guide through all of this. We also have quite a few researchers, including Subina Mehta, Monica Crook, and Catherine Doe, who have contributed to this particular project. And we have worked with multiple researchers across the world, either as data analysts, users, or even developers who have developed various tools. We work very closely with the Minnesota Supercomputing Institute. James Johnson and Reed Wagner help us to put in new tools or even update any tools so that they're kind of kept relevant for any of the new workflows that we work in. Again, at various points of this project, we are funded by NSF, NIH, and also the Norwegian Centennial Chair. If you're interested, you can go to this website to know more about Galaxy P, or you can follow us on Twitter if you're interested to know more about the latest manuscripts or work that we have been doing. With that, I'll be happy to answer any questions if there are any. Thank you.
Thank you, Pratik. Anybody have any questions? I was wondering, Pratik, if you could just give a minute or two overview of how somebody that might want to start using Galaxy P, what that process looks like.
Right. So, whenever a new user, either at the university or from outside the university, has come to us, we have most of the times directed them to, we basically have a conversation with them in terms of understanding what the project is about. And then we direct them to the right tutorials that are available on Galaxy Training Network. The Galaxy Europe resource, or the Galaxy Europe web server, is a pretty good resource wherein most of our workflows and tools are available, and it has a decent amount of memory or storage that one can use to perform uh, project analysis. So we that's what we have been doing within the university. Sometimes, most of the times, we do it on the local server at the Minnesota Supercomputing Institute. And again, as issues come up, or if there are any new outputs that a researcher would like to be interested in, we definitely are in touch with them and help them out. So um, definitely a conversation followed by directing them to the right resources, and then helping them as the workflows are run. I mean, our goal is mostly to ensure that most of the times the researchers are the ones who analyze the data, but there could be times wherein we could actually help them to add some new modules to this analysis as well. Yeah.
I have a question, if you don't mind.
Sure.
So you were mentioning, at least with one of your metaproteomics experiments, having maybe a little bit of difficulty discerning between bacterial proteins versus eukaryotic host proteins. That's actually something I've been kind of considering in some experiments, or how to even kind of tackle that potential issue. Do you have any recommendations, or are you working on or I guess any recommendations for kind of teasing that apart when it's not possible to get a super clean, say bacterial population or host population, and subtract out the contribution potentially of the bacterial proteome?
Yeah, I mean, most of the time we rely on this tool called Unipep to kind of give us a pretty good indication about, whether it's of bacterial origin or of a host origin, right? But in this particular case, the 20 peptides that I showed you were actually it's quite interesting, because some of these peptides seem to be either associated with the variable portion of uh, of the immune system, or if you go down the list, when you're doing the blast P analysis, you can sometimes see bacteria present there as well. Obviously, in our case, we started, the protein file that we started with was of bacterial origin, so we tend to believe that these are from bacterial origin. But what interested us most was that there was a differential expression when we're comparing it with CF and DC samples, the diseased as well as the cystic fibrosis sample. So for us, even if it is of host origin, I think this could still be very interesting, biomarkers, if we, if they end up being seen in the targeted analysis as well. But if we come through such such a in such cases, what we basically do is we then start using other tools such as BlastP or Megan and other tools, basically put it through orthogonal analysis to ensure that we kind of try to find out which particular whether it comes from bacteria or host. But in most cases, we actually do not see that as a problem. But this was kind of a unique situation wherein we found that they're differentially expressed, and looks like they come from this immune system, wherein there is a lot of variable regions, and hence, but that's something we will kind of keep working on. So our suggestion is to either use multiple algorithms to find out more about it, or design experiments or follow-up experiments wherein you can find out whether they're indeed of host origin or bacterial origin.
I see.
Yeah,
Thank you. Yeah.
Other questions? Pratik, I was wondering if you would comment. There's a lot of different data acquisition methods out there, and it seems like you've been through data analysis workflows on many of them. Do you have any consensus on what the right combination of DDA, FAMES, DIA is at this point for maximizing coverage and or quantitation of metaproteome samples?
So most of our data analysis has been on the DDA samples, because that's what was available earlier. FAMES and DI have have just started becoming more accessible now, and the initial data that we have got seems to be very promising. I mean, it could be possible 3 years from now if you were to do quantitation, and if you have a choice, DIA could be the choice because it really gives a really nice, very less missing values, good quantitative reproducibility, and depth of coverage and all of that. But we would like to use it on at least 2 or 3 data sets and ensure that. And we're actually doing one more experiment right now wherein we're looking at a mock microbial community wherein we know the amounts of bacteria that are present, and we would like to see how well DDA does as compared to, or how well DI does as compared to DDA, as compared to the the known amounts of the sample. So once we have some information there, and maybe after doing some more functional analysis studies, that could be the way to go. We haven't really used much of FAMES data, except that it really helps us to get deeper into the sample. In the sense, that's one data that we have used that I showed just now. There's also opportunity to look at ion mobility data sets, but we haven't handled those. So there's a lot that can be done. It's just that we'd like to test these before we can confidently say, yes, that's the way to go if you really have an access to these instruments or acquisition.
Makes sense. Any other follow-up questions from the audience? Well, thank you again, Pratik. I did